Good afternoon, students. So, today we will start our topic by taking up some more patterns of inheritance. So, today we'll talk about pleiotropy. We have already discussed about direct dominance and recessiveness, and then incomplete dominance, codominance. Now, what is this pleiotropy? Pleiotropy means when one gene affects multiple characteristics. That is, the same gene is affecting first feature, second feature, third feature, maybe. So, when more than one character is affected by same gene, that is what is known as pleiotropy. Here you can see the definition of pleiotropy. So, pleiotropy describes the genetic effect of a single gene on multiple phenotypic traits. The underlying mechanism is genes that code for a product that is either used by various cells or has a cascade-like signaling function that affects various targets. So, what do we understand by this? You see, the genotypes considered so far, whatever we have discussed, they have a specific effect on a specific trait. But this is not true for all the genes. Probably most genes, they influence not only the traits generally associated with them, but also some other traits. So the multiple effect of a gene is what is we call as pleiotropy. That means more than one effect. And a gene having multiple phenotypic effects is known as a pleiotropic gene. Now, pleiotropic genes may not have equal influence on all the traits they control. A pleiotropic gene may cause a very evident expression of its specific trait, which we can call it as a major trait or the major effect, and a less evident expression of its other traits known as the secondary effects. Clear? So, if we talk about the examples of pleiotropy, before I discuss this example of the grain, let me tell you some of the examples. That is the genes that control the flower color in sweet pea. They also control the color of the seed coats and the red spots in axils of the leaves. In cotton, a gene for lint also affects the plant size, ball size, seed number, right? Another example is of Drosophila. The gene for wing size also affects the nature of balancers, eye color, fertility, longevity. One more example of pleiotropy is the human disease known as sickle cell anemia. The genes which cause this disease alter the type of hemoglobin and also change the form of red corpuscles. Right? So, in detail, we will take up one example which is given in your books, that is the starch synthesis in pea. Although we see the pattern is somewhat like multiple allelism, but it is not controlled by the multiple alleles, right? It is controlled by a single gene having two alleles. So, starch synthesis in the pea plant is associated with the phenomenon of pleiotropy, right, and incomplete dominance also. So, the gene associated with starch synthesis controls the size of the grain formed as well as its shape. So, when we look at the F2 generation of this cross, where heterozygote individuals they are crossed, we observe the results of the offsprings of the two different characters, that is the shape and the size. Here, if we take as dominant gene as the capital R and the recessive gene as the small r, then we see capital R and R as round, then heterozygote also round, and the double recessive as wrinkled. But if we look at the size of the grain, then capital R and R forms large grains, large starch grains, whereas capital R and small r again forms the intermediate size of the grains, these starch grains, right? Whereas the small r and small r, that is the double recessive, it will form small ones. You see the difference in both the characters? So here what we see is that the same gene R affects the two characters in different ways. The capital R gene shows dominance over the small r when the shape is being decided, but this capital R is however incompletely dominant over small r when it comes to the size of the grain. So the difference in the size of the pea grains, they can be understood as the gene product, that is the starch is not synthesized in the same quantity in all the three genotypes. The heterozygotes produce somewhat less starch than the homozygous, and this results in the difference in the size of the grain, right? So here you see that the gene decides two different things, that is the same gene is deciding the shape as well as the size of the starch grain. So if starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance, right? So dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene. So what do you understand here? You can see here in this cross involved here. In the F2 you see the gametes formed are capital R and small r, and the resultant these four different types, as you have already discussed in the monohybrid cross. Like this you see the representation is the same, but the gene is also the same, but the expressions for two different things are different. That is for the round and the wrinkled shape of the seed, and the grain, starch grain size here differs, right? So the inheritance, that is the size is like that of dominant and recessive, but that of the starch grain is like that of incomplete dominance, and two effects are being shown by the सेम जीन, सो इट इज़ प्लियोट्रॉपी. तो जब एक जीन से हमारे को एक से ज़्यादा फिनोटिपिक इफ़ेक्ट्स मिलते हैं, दैट इज़ व्हाट इज़ नोन एज प्लियोट्रॉपी. और जो जीन इस इफ़ेक्ट को शो करती है दैट इज़ अ प्लियोट्रॉपिक जीन. प्लियोट्रॉपी जो है ये कन्वर्स है पॉलीजेनिक इनहेरिटेंस का, जिसमें क्या होगा, पॉलीजेनिक मीन्स मोर देन टू. वी विल टेक अ पॉलीजेनिक आफ्टर दिस. प्लियोट्रॉपिक में जीन एक है, एक्सप्रेशन एक से ज़्यादा वो दे रहा है, फिनोटिपिक इफ़ेक्ट्स उसके जो हैं टू और मोर देन टू हैं. वेयर एज इन द पॉलीजेनिक इनहेरिटेंस there are two or more genes which have an additive effect on a single phenotype. You see, they are both converse of each other. In pleiotropy एक जीन है, phenotypic effects एक से ज़्यादा हैं, whereas in the polygenic inheritance genes दो या उससे ज़्यादा हैं लेकिन उनका इफ़ेक्ट एक चीज़ के ऊपर है, सिंगल phenotype के ऊपर. Okay, clear up till here? So now we will take up the next thing, that is polygenic inheritance. जो आपको डिस्प्लेड दिख रहा है अपनी स्क्रीन्स पे, ये क्या है, कैन यू टेल मी? This is almost the bell-shaped graft which we see in the skin color inheritance of humans. Very fair to you see the intermediate coloration with the peak and then coming towards the very dark. सो ये पॉलीजेनिक इनहेरिटेंस का एग्जाम्पल है, जिसमें ये वेरी लाइट स्किन कलर इज चेंजिंग टू डार्क 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 अप टू द डार्केस्ट। और सबसे ज़्यादा प्रोपोर्शन पॉपुलेशन का कौन सा आता है? इंटरमीडिएट वन, राइट। सो नाउ we'll take up the polygenic inheritance. So polygenic inheritance occurs when one characteristic is controlled by two or more genes. Often the genes are large in quantity but small in effect. Examples of human polygenic inheritance are height, skin color, eye color, and weight. Right? Here you see the example of skin color, in which there are these three genes involved, A, B, and C. So obviously we represent the dominant characteristic by a capital letter and the recessive one by a small letter of the same alphabet. So we see the A, B, and C in capital letters are the dominant genes, whereas small a, b, c are the recessive genes. So dominant genes show darker skin color, whereas the recessive genes, they show the lighter skin color. So on your screens you can see the number here. It is representing the number of the genes. Here you see capital ABC, on the other side again capital ABC. These are the gametes formed by the male and the female parent. So here you see it shows how many dominant genes are present. The number 6543 shows the number of dominant genes present by the combination of these different gametes. Right? So in the last column, if you see, that is small abc and small abc, and it's written 0 there in the extreme right down. And this means no dominant gene is present, and this is the lightest skin shade. Where 1, 1 are present, it is slightly darker than this one, and then 2, and then 3. Now this is the bell shape graph which you see on the right side. This shows, this graphical representation or the bell shape shows the number of different skin shades from lightest to intermediate maximum number, and then again coming down to the darkest one. So the lightest and the darkest are on two extreme sides, and you can see they are minimum in number, whereas the intermediate color is maximum in number. This is the frequency, the graphical representation which you can see. Here now you can see the cross with the three genes showing. This is the same thing shown in a slightly different manner, starting from the parental generation and then coming to the gamete formation F1 generation, and then coming down to the F2 generation. And the proportions shown here, there are 64 different blocks here, and of this the proportion is shown here of different skin shades, that is 1 by 64, 6 by 64, 15 by 64, 20 by 64, again 15 by 64, again 6 by 64, and last darkest again 1 by 64. So this is the proportion from the 64 different combinations of different skin shades which are formed. Clear? So now we come down to the next part, that is the chromosomal theory of inheritance. Now what is chromosomal theory of inheritance? You are aware about chromosomes, although at the time of Mendel no concept of chromosomes, genes was there. But now we are very well aware about chromosomes and genes. So we know that the chromosomes and genes are present as pairs in diploid cells. Homologous chromosomes separate during meiosis. the type of cell division which occurs for the formation of gametes. Fertilization restores the chromosome number to diploid condition. The chromosomal theory of inheritance claims that it is the chromosome that segregates and assorts independently. Clear? So these are the points. Now, what does it state? So the chromosomal theory of inheritance proposed by Sutton and Boveri states that the chromosomes are the vehicles of genetic heredity. Neither Mendelian genetics nor gene linkage is perfectly accurate. Instead, chromosome behavior involves segregation, independent assortment, and occasionally linkage. So, here in this picture, you can see the chromosomal theory of inheritance with an example of the yellow and green seeds, round and wrinkled seeds on the chromosomes, the example which you have discussed for the dihybrid cross of Mendel. Now here you can represent it by the chromosomes. So it is represented here, the chromosomes are representing it. You see here the YY and the R and R chromosome. This is a hybrid capital Y, small y in the homologous pair of the color of the seed, and whereas the round and the wrinkled shape of the seeds in the next homologous pair is capital R and small r. So for division before meiosis, the chromosomes they will replicate, and here you can see the chromatids which are formed. After that, in meiosis 1 is the reductional division. So these two cells are formed as a result of meiosis 1, and the chromosome number here is reduced to 1/2, and then as a result of meiosis 2, 4 cells are formed. And you can see their assortment, the independent assortment which is formed, right? So we can compare between the behavior of chromosomes and genes. Both occur in pair, right? Now you can, by going through this table, you please decide now that what represents what is chromosome, and the from A and B column what represents the chromosome and which one represents the gene. I hope you will be able to do that. So go through it once. So I hope you were able to decide. Now here you see again the same representation of possibilities, that is first possibility and the second possibility in during the segregation. So here the chromosomes they are representing like one long orange and short green chromosome, and the long yellow and the short green chromosome at the same pole, whereas one long in the possibility 2, one long orange and short red chromosome, and long yellow and short green chromosome at the same pole, right? So here you see orange and green, yellow and red, orange and red, yellow and green. Then during meiosis, what happens? They separate, and the ultimate results of independent assortment which are formed in possibility 1 and possibility 2. So by taking these 4 chromosomes, you can see the net results in the germ cells, the 2 possibilities of the combinations which can be formed. Right? Different colors are given to show the different alleles which are being inherited, and the chromosomes to show the representation of different chromosomes. So this is all for today. So any doubts you can ask, and next class we'll start with the linkage and recombination. Till then, please stay safe and stay healthy.